The Tasmanian devil, Sarcophilus hare, is the largest extant carnivorous marsupial. The species became extinct on mainland Australia around 3,000 years ago and is presently found only on the island state of Tasmania. Current status of the Tasmanian devil is endangered as stated in UCN Red List. Last assessment for the UCN Red List was in 2008, so it may be worse than this. There are 3 known transmissible tumors in vertebrates, but 2 of these have occurred in Tasmanian devils. One apparent factor here is a predisposition to cancer. Another seems to be the loss of T-cell diversity in devils following 2 years of age, which would explain the inability to reject Dftd as an allograft, and the fact that Dftd mostly occurs in adult devils. Dftd cause live cancer cells transmitted as an allograft by biting during aggression and breeding. Infected individuals show ulcerated lumps of soft tissue around the mouth that prevent feeding. Infection caused death within 6 to 12 months of infection in almost all cases. Dftd associated with declines of up to 89% in the population. The predisposition for cancer coupled with anthropogenic pressures present a clear threat to the long-term persistence of Tasmanian devils in the wild. Tasmanian devils with large facial tumors were first noticed in northeast Tasmania in 1996. A second, distinct lineage of transmissible tumor was identified in 2014, and these are now known as Dftd1 and Dftd2 respectively. Between 1996 and 2015, these 2 transmissible tumors wiped out 95% of devil populations in affected areas. Dftd1 is female-originated from diploid cells which causes extinction of local populations with 95 to 100% mortality rate. On the other hand, Dftd2 is male-originated from tetraploid cells and has lower mortality rate as its impact on the population is still unknown. First management strategy is vaccinations, which has been done a few times. Although the disease is thought to avoid immune detection, recent findings indicate that the species are surviving for longer with the tumor and experiencing immune responses to the tumor, which may make a vaccine more feasible. Development of vaccines for cancer is a very complex process. First, some captive devils have been induced to elicit an effective immune response against Dftd1. Then use of irradiated Dftd1 cells but failed to induce cytotoxic or humoral immune responses. And was on small sample size, only 6 devils. 3 produced anti-Dftd1 antibodies in response to immunization. 2 of them produced low titers of antibodies which did not stick around for long. The individuals with an immune response lasted 189 days without getting tumors after being exposed and then developed small tumors. These were surgically resected. So, devils can produce specific antibodies against Dftd1, but this is variable between individuals and not lifelong. Vaccinations using MHC class I-positive Dftd1 cells are more effective than using negative class cells and can be used as immunotherapy in devils. With mass vaccination and an effective vaccination, then this vaccine likely to be very successful. However, for this to happen we need an effective vaccine and delivery method, that is funding, within the next few years, which appears unlikely. There is little studies on vaccines and mostly done on small sample sizes. Trials as low tend to use older devils and may need to be euthanized, age-related health issues before results can be collected. Wild Devil Recovery Project by the Tasmanian government in 2015 used vaccines on 52 younger devils from disease-free populations before being released and given immunization and monitored. 19 devils released in first trial, which got 2 4-monthly vaccines and then booster vaccine at 5 months post-release. No significant difference in the levels of antibodies detected between the 2 cell types, and average levels of anti-Dftd1 antibodies increased after each subsequent vaccine. No real ethics issues here unless limited vaccinations and need to select individuals to vaccinate. Vaccines potentially solves the issue either by reducing severity of disease so it is not fatal, or by reducing risk of getting the disease. Another potential management strategy is to identify Dft-resistant devils and attempt to spread the resistant alleles into affected populations. This is potentially a long-term solution. Populations in the west of Tasmania, which is currently uninfected, are weakly genetically differentiated from those from the east coast. The different patterns of disease progression and demographic effect in northwestern Tasmania, the detection of antibodies for devil facial tumor disease in individuals at West Pennsylvania, and knowledge of the genetic population structure of devils at both neutral and functional markers involved in tumor recognition suggest that there may be a genetically based resilience to the disease. If resilient populations could be established in parts of eastern Tasmania by translocating resistant animals, these animals may help repopulate currently very small eastern populations. However, differences in microsatellites which reflect functional MHC differences and whether such differences would be sufficient to make some West Coast animals resistant is still unknown. Further, it is possible that the tumor itself might evolve in an adaptive fashion, so that even if some West Coast genotypes are currently resistant to disease, this might not be the case in the future. Moreover, the mechanism for resistance and whether the resistance phenotype would have similar effects in other outbred populations is unknown. Additionally, translocation among wild population risks introducing Dftd strains with higher virulence due to the long latent period of Dftd, which can extend for at least 13 months in some cases. For example, Dftd strain replacement has been documented to result in a rapid increase in disease prevalence, population decline and reduced mean age of the population. This technique has not been adopted into devil conservation efforts primarily because, 1. It is a relatively new concept with some aspects still speculative. 2. Investigations into genomic regions associated with Dftd resistance are still under investigation, and 3. There are concerns that translocating individuals selected specifically for disease resistance may increase adaptability towards the disease or reduce future adaptive potential. Another approach is disease-free breeding projects in captivity. Already effective with more than 490 disease-free devils in over 20 institutions across Australasia. However, cannot release these populations because they would come into contact with diseased individuals and likely contract it. This would leave others to die by not trying to find a cure, not actively harming any individuals. This won't cure disease and thus not solving the problem, and the disease could re-emerge. However, this would solve the issue of number of individuals declining but not in real-life setting and only in captivity, and thus not really conserving it. There is also selective culling. One study model suggested that 96% of infectious devils would need to be removed on a regular basis to stop the disease. This is unlikely to be possible as devils are hard to trap and after infection the tumors take time to grow. Clinical signs are not immediately obvious, estimated around 6 months. Reduces transmission through reducing population density and increasing the removal of infected individuals and thus removing opportunities for infection. Likely to be successful in that there is no known reservoir, so probably fairly simple to remove the infected individuals and have the infection occurrence reduced. However, one study found no evidence that selective culling of infected individuals prevented the disease spreading, no decreased rate of disease progression or population impacts. It has been trialed in 2.5-year study on the Forestier Peninsula, has not displayed a decline in prevalence and population has shown no sign of recovery. A very high removal rate is required if this is to be successful, thus this would be expensive and labor-intensive. Killing individuals. The general public generally find this unacceptable, although they are going to die anyway, as it is painful. Devils can be infectious for 6 months before showing symptoms, so will not solve the problem on its own, as not all infected individuals will be removed. Removing numbers from an already diminishing population without implementing measures to increase it is not the best approach. Lastly, doing nothing. 100% effective in ensuring species become extinct, which is not what we want. Allowing an entire species to go extinct without attempting to prevent it is not ethical at all. Disease may mutate and be able to jump into other populations and could be devastating for more than one species. This can remove the species and has cascade effects onto other species.